So related to, what we've talked about in the motor control section, the spinal cord is actually kind of interesting, even though it's not going to be on your exam, on your lab exam. But here you can see the dura mater continuing out through the spinal cord. And like we talked about in the brain, the arachnoid is adhering to the surface of the spinal cord. And then the pia would be underneath that. But you can see the spinal rootlets coming up. And, when we talk about spinal nerves, we often think of a single nerve coming out at each vertebra. And in reality, as you can see, they're really a series of rootlets. So one spinal segment is a lot of little nerve fibers coming out. And on the dorsal side, a lot of sensory nerves going in. And they collect at the meninges and then go through the meninges as the spinal nerve. So here you can see the spinal nerve for each segment coming out. And that's a collection of all those little rootlets. And you can see this lump, that's the dorsal root ganglion.
So when you say like a bunch of these little nerve rootlets, so like within one little rootlet, is there a bunch of nerves in there, or a bunch of axons?
That's a bundle of axons. Each of these little rootlets is a bundle of axons.
And so then they,
in this case, coming out of the ventral horn, coming from the ventral horn in that particular region. And one spinal nerve would include the rootlets of all of these little bundles of fibers coming out.
Okay.
And also you can see that the spinal nerve, or the spinal cord, is pretty short and doesn't go all the way down to the end of the vertebral column. So your spinal cord stops developing early on in your postnatal, and the vertebral column continues to grow as you grow, as your body axis grows, especially through puberty and your growth spurts, etc. And so the spinal nerves that would come out at each of these segments are pulled down as the vertebrae grow and as those intervertebral foramina move down further. And so these are all spinal nerves that are continuing down in the vertebral canal to come out at the lumbar and sacral as the lumbar and sacral spinal nerves. And this is called, because of that appearance, this is called the cauda equina. And the cauda equina is what you'd see in the vertebral canal below about L1, I think, is typically where the spinal cord stops. And where it stops, that's called the Conus Medullaris. And that's the end of the spinal cord. And then below that is all just spinal nerve roots that are going to come out at more inferior foramina. And so that's where you would take cerebrospinal fluid from, or where you would make an epidural injection, is down below that, because then you don't have to worry about damaging spinal cord tissue, because it's all canal that just has these little roots going down it. Mm-hmm. So that's kind of interesting to look at the spinal cord and see how it really looks.
Yeah, why don't we talk about that in life?
In life?
Yeah, like I feel like nobody talks about that. Nobody told me that the spinal cord.
Look at the brainstem and the cranial nerves. One of the things that's interesting about the human brainstem that makes it really tough to identify things on the ventral surface is that we have this flexure right about the mesencephalon and the diencephalon, where the axis goes from vertically oriented to anteriorly oriented. And so the problem with that is, for us, looking at structures, is that stuff in the midbrain gets buried in there, but we can still pick apart some of the things that we've seen on the sheep brain stem. So if we look at the cranial nerves, here you can see the olfactory bulbs at the end of the olfactory tract that run out ventral to or inferior to the frontal lobes. And so one thing I'll point out here, when we talk about the orbitofrontal cortex, you can see where the orbits are. where the eyeballs and the other structures in the orbit would be. They're right out in front here. And so this is what's called the orbitofrontal cortex. And we'll talk more about that in some of the higher processing stuff. And so the optic nerves are going to come out toward the orbits, and the three nerves that innervate the extrinsic ocular muscles also will run out that direction. And so here you can see the optic nerves going back to the optic chiasma, and then that splits, and like on this one you can see the optic tracts continuing laterally that are going to wrap around the thalamus. You can also see here the infundibulum to the pituitary. So see this stalk right here? That's the infundibulum, and then the pituitary would sit right in here, which is removed for reasons that I talked about before. On either side of that, you can see the oculomotor nerve that's coming out, as we saw in the sheep, coming out of the floor of the mesencephalon, or the tegmentum, and is also going out to the orbits. And on these, because of all the other structures in the way, it's really tough to identify the trochlear nerve, which is also going to come out from the midbrain and then wrap around and go up to the orbit as well. But the trigeminal has the same general orientation that we saw in the sheep. So you can see, as you look at the floor of the brainstem, you can see the big pons here, and you can see the size of the pons here. And like I talked about yesterday, this bundle of fibers, the brachium pontus or middle cerebellar peduncle, is a huge tract of fibers, and the area in the pons that are generating those fibers, the pontine nuclei, is a large area as well. And so in here, the pontine nuclei are giving rise to fibers that cross the midline and form the brachium pontus on the opposite side, the middle cerebellar peduncle on the opposite side. And so in your sheep brain, they talk about transverse fibers of the pons, and those are the fibers that are coming from the pontine nuclei and forming the middle cerebellar peduncle.
Those aren't talking about like the transverse fibers from the trapezoid body?
No,
Okay.
they're the transverse fibers of the pons.
Okay, different.
And the trapezoid body's back here. And so you can see the size of this in humans, associated with the cortical input that's coming to the cerebellum. And we'll talk more about that next week. So the trigeminal nerve, like in the sheep, is coming out basically in the middle of all those fibers. It penetrates out. And just like we saw when we looked at the sheep brain, that little stub of the trigeminal is pretty much in the center of, or near the center of, the brachium pontus. And then caudal to that, as we move back for the cranial nerves, you can see the facial nerve is still a pretty long stump here. And it's coming out just caudal to the trigeminal, and unfortunately the abducens, which comes out just caudal to the pons, is kind of torn away on this side, but you can see little fibers of the abducens here on the opposite side. So the abducens is going to collect and run up to the orbit as well. So you've got the optic nerve going to the orbit through the optic canal, and then you've got the where'd the oculomotor go, and the trochlear and the abducens all running through the superior orbital fissure to the optic the space to the extrinsic ocular muscles. The vestibulocochlear also is much more prominent in the human specimens, and so we can actually see a branch to the vestibulocochlear coming out just ventral to, or in our case, anterior to the floccular region of the cerebellum, so just underneath the flocculus, as I talked about here, but usually we can only see a little tiny stump if it's at all visible that comes out just ventral to the flocculus, or floccular nodular lobe as they identify it there. And so that's 6 over here, then 7 and 8, and the 9th and 10th, I've mentioned that they come out as a series of roots, and usually the first root is glossopharyngeal, maybe the first couple, and then there's a series of vagal roots coming out, and they come out dorsolaterally off the brainstem, the medulla, and you can see those roots here all lined up, and those are all glossopharyngeal and then vagal roots that come out. The spinal accessory we can see along the lateral surface, and you get a really good view here of these bundles of fibers that are coming up from the spinal cord, and they're picking up rootlets in the spinal cord, and then they pick up a few more rootlets in the medulla, and then they'll collect and go back out the foramen magnum to the nerves in the upper shoulder and neck, or the muscles in the upper shoulder and neck. And ventrally, move the vessels over here, we have these nerve fibers that are coming off the floor of the medulla, and those are the hypoglossal nerves. And the hypoglossal nerve is going to come off in this ventrolateral sulcus here and go out to the throat, the lungs, the tongue, and the muscles, the hypobranchial muscles. One other thing that's more obvious here that you can't really see in the sheep brain are the olivary complexes, and you can see the olives protruding lateral to the pyramids, or the pyramidal tracts here. And these olivary complexes are the inferior and superior olivary nuclei that we've talked about. So the inferior olives we'll talk a little bit more about next week, participate in the cerebellar control, and the superior olives we talked about with the auditory system and the medial and lateral superior olivary nuclei. And that's something that isn't very evident on the sheep. Now, one of the things that I like about these, it makes it tougher to identify the nerve fibers, but the vasculature in here is still intact. And you can see what's called the circle of Willis. And it's not something we talk about in our lab, but it's really pretty cool. And that's the blood supply to the brain. And so we get blood supply from 2 pairs of arteries. From the spinal cord, we have the vertebral arteries that run up through the cervical vertebrae and go into the foramen magnum and run across the floor of the medulla and then join about the midbrain, the pons and the midbrain, and form the basilar arteries. And so these are the vertebral arteries. They come together to form a basilar artery, and then that splits to go around the pituitary. And it joins up with, it connects, see these arteries right here, they're kind of cut, but they connect that basilar artery with these 2 large arteries that have been cut in cross-section. Those are the internal carotids. So your carotid is going up into the floor of the brain, the cranium, and it continues around the brainstem and goes up into the cerebral hemisphere as the middle cerebral artery, and supplies blood to a lot of the core structures in the brain and a lot of the cortical structures out here. And so that's an important artery because that's often where strokes occur or bleeds occur if you damage the brain. And it will produce damage to the parts of the cortex that are in those regions. Now, the internal carotids also have a branch that continues around the optic chiasma here, and you can see this branch, and it forms the anterior cerebral arteries, which go up between the frontal lobes. And they're connected by a little arterial bridge called the anterior communicating artery. And that communicating artery, in combination with these posterior communicating arteries, form a circle. And what that means functionally is that if you block blood flow in one of those vessels, the blood can still get around to that part of the brain by going through that circle of Willis. And so it offers a redundancy where you have blood supply through the carotids and the vertebrals that allows blood supply to the brain even if there's some kind of impingement on one of the arteries, which is, it's pretty cool how that works. And you guys might talk about that in lecture, in anatomy lecture, when you get to the vasculature of the head. Now, the other thing I wanted to talk about is just a little bit, because we're in the process of talking about a lot of structures in lecture, that are in the cerebral hemispheres or that involve pathways up to the cerebral hemispheres. And so I wanted to just kind of give you a view of what this looks like, those structures look like in the human, and reflect on what we see in the sheep. And so in this one, it's a little off center, but similar to what you've seen in the sheep already. Here's the midbrain, here are the colliculi, and they sit in the tectum, the roof of the midbrain, and they sit right above the cerebral or the mesencephalic aqueduct. And so this is the tegmentum of the midbrain. So when we talk about substantia nigra or the red nucleus or the ventral tegmental area, those are all nuclei that are located here in the tegmentum. And when we talk about the superior colliculus and inferior colliculus, those are little bumps up here. And you'll notice that in humans, they're relatively much smaller than they are in most other mammals. And that's because a lot of their visual and auditory processing is very simple reflexive processing that's done in the midbrain. We do a lot more with the cortex and the combination of the cortex with the basal nuclei and the cerebellum to adjust or move our eyes or process auditory information. And then here is the diencephalon, and you can see the mammillary body here. You can see the infundibulum. It's the pathway that has an extension of the third ventricle, which connects to the mesencephalic aqueduct. And this is the anterior commissure, which is much larger in these guys, in humans. And if you remember, the anterior commissure is a little bundle right here. And that separates the telencephalon from diencephalon. So this is hypothalamus down here, thalamus, and then unfortunately the pineal has been popped out of this guy, but it would sit right up here. And the third ventricle communicates with the lateral ventricle through a little gap that goes underneath the fornix. So this is the fornix, but it was cut offline and the rest of the fornix has been removed, unfortunately. And this is the interventricular foramen that connects the lateral ventricle here with the third ventricle. And if you look in the lateral ventricle, you can see on top of it is this big, thick corpus callosum. And you can actually see the bundles of fibers that are coming out towards you in this. And underneath that, on the floor and on the lateral wall of the lateral ventricle, is the caudate nucleus. And so the basal nuclei are right down in here, the basal ganglia that we've been talking about. And in this section, where I've removed most of the parietal lobe and all of the occipital lobe, you can see the caudate nucleus pushing out into the lateral ventricle here. And what you can't see in this one is where the fornix comes up and forms the fibers that connect the hippocampus to the mammillary body and to the septal region. or basal forebrain. And those are illustrated here. So here's the fornix here. And this is the body of the fornix. And as it goes down, it forms the columns, which are just in front of the third ventricle. And then those columns split at the anterior commissure. So they would split right here. And the post-commissural fibers go back to the hypothalamus and the mammillary body. And the pre-commissural fibers go to the septal region in here, as they do here. And you can't see those fibers, but they come down like this. And here's the mammillary body and the sheath. And then as they go back, you can't see it on the human specimen, but the hippocampus is underneath this bulge and is producing this bulge. And that surface of the bulge are fibers, a thin sheet of fibers called the alveus. And those collect here cranially and laterally as the fimbria. So when you're looking at the coronal sections, you'll be able to see the alveus on the surface of the hippocampus, and the fimbria as white matter that extends out laterally from the hippocampus. And those fimbria collect to form the fornix as they go forward. And then again, as you follow that out into the temporal lobe, the hippocampus forms this C shape. So you'll often see it in the coronal sections, either cut through this long part of the C, the caudalmost part of it, or you'll see two areas of hippocampus and fimbria. And then at the end of the hippocampus is a little bulge, the amygdaloid body or amygdala. And those, unfortunately, I don't have a section that goes through those parts of the temporal lobe, but the amygdala would be down in here, and then the hippocampus is coming down here and going up into the parietal. And the other thing I wanted to point out, and I think I mentioned this last week, but if we move the hippocampus out of the way, you can see the colliculi, the midbrain, the tectum here, and the thalamus, at least the caudal thalamus. And in the caudal thalamus, you can see this bulge that protrudes dorsally. That's the pulvinar nucleus that we haven't really talked about, but plays a role in relaying information to the more complex information to the cortex. And then at the top of the optic tracts, those terminate in this bulge that is dorsolateral, and that's the lateral geniculate body or lateral geniculate nucleus. And then the medial geniculate nucleus is just below that, and that's the auditory relay that goes up to the auditory cortex. And so you can see those in this structure, and you can relate those. If you look at the coronal sections, they'll show you each of those nuclei in different of the plates in the atlas. And then what else did I want to talk about? So I think it's valuable to be able to picture these because they aren't linear and because they curve. So when you're thinking about the fornix and how it's oriented, how it curves around, and you can see the fornix, you can see the body of the fornix in some sections, and a little bundle of the fornix that's going back in the hypothalamus. And that's because it forms this backwards C shape that goes back to the mammillary bodies. And when you're looking at the caudate and the tail of the caudate, that extends back in a forward C shape, just like the hippocampus and the fimbria do. But they're on opposite sides of the lateral ventricle. So when you look at the coronal sections, it's important to be able to think of that orientation.
What shape does the putamen make?
It's much shorter than the caudate. So the caudate extends as a tail back to about here. The putamen and the globus pallidus are located up in this anterior portion of the basal nuclei. So in the sections that you have here that are in front of the, or cranial, or anterior to the, like the anterior commissure, you'll see the caudate dorsally, and then the putamen ventrolaterally, and the globus pallidus ventromedially. And you can't see any of that in these, but in the coronal sections you can. So they're boom, 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 like that. And one of the things to notice when you're looking at the basal nuclei, the putamen is more lateral and it's ventral to the caudate. And it is usually a real clean nucleus. So it's really blue in the sections that we look at. It would be pretty clean gray matter.
And like internal and external capsule.
And then the internal capsule goes down between that and the globus pallidus. And the globus pallidus has more fibers running through it. And so you often see a patch of, or patches of fibers and cells in the globus pallidus. And then those are enclosed in the external capsule. And then just outside of that is the claustrum, and that's enclosed between the external capsule and the extreme capsule. And each of those capsules are bundles or sheets of fiber.
Okay.
Okay.